Good day, my life science student, Mr. Hescox here. I was going to let you guys work on these Punnett squares, which is a huge part of genetics. We use Punnett squares to predict the allele combinations of offspring. So what I thought I'd do is make a little video. I know that some people are at home, or you may just need some extra help with this. So, is there a black sheep in the family? In sheep, the gene for white wool is capital W. So anytime you see a capital letter, that's a dominant allele. The gene for black wool is lowercase w. The sheep in the P generation, which stands for parent, produce the sheep in the F1 generation. That actually stands for first filial. You can think about it as like first family. The F1 sheep are the parents of the F2 generation. See the drawings below. Write the genes for wool color. Also color the sheep white or black according to their genotype. So for this, genotype is right here. This one has white and white. It's what we call homozygous, and it would be white. This one here is not white. Oops. This one, little w, is black. The other little w is black. This is homozygous recessive. So this sheep right here would be black, and then you'd color it in black. If we move down, you've got a cross between these sheep. So when you draw your cross, you put, this one's going to be capital W on one side, little w on the other. Now you want to be very careful when you set up Punnett squares. The sloppier your handwriting, the more dangerous this becomes. So you bring one down, bring it over, bring it down, bring it over, bring it down, bring it over, bring it down, bring it over. Now these have one tall, one short. These are what we call heterozygous. But because the white is dominant, all of these are white. In fact, this one is 100%. White. And in here, you would write capital W, little w, capital W, little w. You guys are going to do this one down below. Remember that your parents for this one are all capital W, little w, capital W, little w. Okay, so moving on a little bit further. What color is the pod? Green, capital G, is the dominant color of pea pods and pea plants. Yellow G, lowercase g, is recessive. What are the phenotypes? The phenotype is what you see. The genotypes are the types of genes, like literally the type O genes. So big G is the alleles. So that's dominant, dominant, and then the dominant color for this one is green. Here, big G, little g. Well, big G is dominant over the little g. The dominant color is green. The other color is recessive, so this one will also be green. Alright, so fill in the Punnett squares below to show the outcomes of the crosses. Next to each genotype, write the correct phenotype. So we're going to set these up right now, and then we're going to actually add the ratio too. It's called the phenotypic ratio. So here you have big G crossed with big G, little g. Make sure that one parent is always on top, one parent is on the other. You never crisscross them. So when I do this, bring one down, big G, bring one over, big G, big G, big G. Bring one down, big G, bring one over, little g. Bring one down, big G, bring one over, little g. So here's green, 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 and green. I would like you guys to write your phenotypes inside each square. And in this case, my ratio for this one is 4 greens to 0 yellows. So if I move down, we're going to jump down and do this last one on this page. Here you have what we call heterozygous dominant and homozygous recessive. So we're going to put the dominant one on top. Capital G, little g. Over here you got homozygous, which means 2 of the same alleles, little g, little g. So you bring one down, capital G, bring one over, little g. Bring it down, little g, bring it over, little g. Bring it down, capital G, bring it over, little g. Bring it down, little g, bring it over, little g. So that is what we call our genotypes. So capital G, green, is dominant over yellow, so this one will be green. Here, I've got a yellow allele, I've got another yellow allele, so this one will be yellow. Here I've got a capital G, lowercase g, this one will be green. Lowercase g, this one will be yellow. Pen's not working very good. So here, you could put that your ratio is 2 to 2, but Mr. Hunter would tell you that that should be reduced, so you divide each side by 2, and your actual ratio for this one should be 1 to 1. 1 green for every 1 yellow. All right, here, this is another old worksheet from a different book that I had. And this is all talking about Punnett squares, phenotypes, and genotypes. So the phenotype is the visible trait or the express trait. And then the genotypes are the types of genes. Basically, it's the letters. So if we look down below, this one's kind of set up for us here. So here we have guinea pigs. You have a black guinea pig and a white guinea pig. Capital B tells you that the black is dominant. So this is a homozygous dominant guinea pig crossed with a homozygous recessive guinea pig. And when you cross them, they all become capital B, little b, every single one of them. So here it asks, what are the phenotypes of the parents? So the phenotypes are what we see. So one is black, and the other one is white. What are the genotypes? The genotypes are always the letters. So in this case, capital B, lowercase b, lowercase b. What are the phenotypes? What do my offspring look like? Well, they're all black. What are the genotypes of my offspring? So that's the letters. The genotypes are always the letters. Big B, little b. So then you'll have these questions here to do. You'll have one that you have to work out on your own. One little tidbit on this one here. It says, what is the probability of getting a black purebred? So pure typically means homozygous. If you hear something called a hybrid, that typically means heterozygous. All right, guys. I think I have enough here that you guys should be able to get this done. Do your best. You can email me if you need help. My email is bmh@blwd.k12.pa.us, and I hope you're well and safe. See you next time.